Hello everyone, this is Sapna from RPIIT Medical and Technical Campus. In previous classes, we have discussed about glycolysis and gluconeogenesis. But in today's video, we are going to discuss about TCA cycle, that is known as tricarboxylic acid cycle and also known as Krebs cycle. TCA cycle essentially involves the oxidation of acetyl CoA to form carbon dioxide and water. TCA cycle is the central metabolic pathway. TCA cycle is the final common oxidative pathway for carbohydrates, fats, and amino acids. TCA cycle supplies energy and also provides many intermediates required for the synthesis of amino acids, glucose, or heme, etc. TCA is the most important central pathway connecting almost all the individual's metabolic pathways. Now we will discuss about the definition of TCA cycle, which is also known as citric acid cycle. Citric acid cycle or TCA cycle or tricarboxylic acid cycle essentially involves the oxidation of acetyl CoA to produce carbon dioxide and water. Location of TCA cycle. And then the reaction occurs in mitochondrial matrix in close proximity to electron transport chain. And here we can see the TCA cycle. In TCA cycle, pyruvate is first of all converted into acetyl CoA in presence of pyruvate dehydrogenase enzyme, and from NAD plus we get carbon dioxide, NADH plus and H plus. Then acetyl CoA combines with oxalate in presence of citrate synthase enzyme. It is converted into citrate. Then water is removed in presence of acetone and citrate is converted into cis acetone. Then cis acetone uh, is converted into isocitrate by removal of water in presence of acetone. Then isocitrate is converted into oxalosuccinate, and this is a reversible reaction in presence of isocitrated anhydrogenase enzyme, and from NAD we get NADH plus H plus. Then oxalosuccinate in presence of isocitrated dehydrogenase is converted into alpha ketoglutarate, and here carbon dioxide is removed. Then alpha ketoglutarate is converted into succinyl CoA enzyme in presence of alpha keto dehydrogenase enzyme, and here NAD plus is also converted into carbon dioxide, NADH and H plus ion. Then succinyl-CoA is converted into succinate in presence of succinate thiokinase enzyme, and GTP plus PI is converted into GTP, and then succinate is converted into fumarate in presence of SDH enzyme, and FDH is converted in FDH2 is converted into FDH. Then fumarate is converted into malate, and here water is removed in presence of fumarose enzyme. Then malate is converted into oxaloacetate in presence of MDH enzyme, and from NADH plus from NAD plus we get NADH and H plus ion. Now we will discuss about the significance of TCA cycle. Complete oxidation of acetyl CoA ATP is generated, final common oxidative pathway. It integrates of major metabolic pathways. Fat is burned on the wick of carbohydrates. Excess carbohydrates are converted as neutral fats. No net synthesis of carbohydrates from fat. Carbon skeleton of amino acids finally enters the TCA cycle. Now we will discuss about the energetics of TCA cycle. 3 NADH gives 9 ATPs, 1 FADH gives 2 ATPs, 1 GTP produces in substrate level phosphorylation. Total of 12 ATPs are produced from 1 acetyl CoA. And then we will discuss about the regulations of TCA cycle. TCA cycle is regulated by three regulatory enzymes: citrate synthase, isocitrate dehydrogenase, alpha ketoglutarate dehydrogenase. And now we will discuss about the inhibitors of TCA cycle. Inhibitors of TCA cycle are fluoroacetate, aristine, and malonate. And here is the end of TCA cycle. Thank you.